Welcome to All About Rx. Today, we are exploring the fascinating world of synaptic transmission and signal transduction. We will break down how neurons communicate across chemical synapses, examine the mechanics of excitatory and inhibitory postsynaptic potentials, and explore how summation, second messenger pathways, and various drugs shape neural signaling. I am absolutely thrilled to guide you through this explainer, because honestly, understanding these microscopic mechanisms is the literal key to understanding how our brains and our entire bodies truly function. We're taking a micro to macro journey today. We'll be covering anatomy of a synapse, excitatory versus inhibitory signals, calculating the grand potential, types of intercellular communication, signal transduction and receptors, and finally, drugs, diseases, and synapses.
All right, let's jump right into section 1, anatomy of a synapse.
At the very core of neural communication is the synapse itself. This is a unidirectional junction, meaning the signal only flows one way. But here's the catch. There is a physical gap called the synaptic cleft. Because this gap is just a bit too wide for a direct electrical spark to jump across, the neurons are forced to use a chemical ferry system. So when an action potential arrives at the presynaptic knob, it triggers voltage-gated calcium channels to open up, allowing a crucial influx of calcium. This calcium influx is what triggers the exocytosis of neurotransmitters into the cleft. These molecules then diffuse across the gap and bind to specific receptor channels on the receiving or postsynaptic cell, which changes its membrane permeability.
Picture the vesicles clustered inside that presynaptic knob. Upon that calcium trigger, they just dump these chemical messengers straight into the cleft. Now that gap is incredibly tiny, literally just 20 to 40 nanometers wide, but it's still wide enough to necessitate this entire chemical conversion process. Once across, the neurotransmitters bind exactly like keys into locks on the subsynaptic membrane receptors, unlocking the next phase of the signal.
But think of it kind of like clearing the cache on your web browser. The chemical message can't just linger in that cleft forever, right? It has to be terminated swiftly so the cell can recover and reset for the next message. The body gets this done in 3 main ways. The neurotransmitter can simply diffuse away, it can be destroyed by enzymatic inactivation right there on the membrane, or it can undergo active reuptake, where transport proteins actively pump it back into the terminal to be recycled.
Moving on to section 2, excitatory versus inhibitory signals.
So what actually happens when those neurotransmitters bind? Well, our nervous system essentially has a gas pedal and brakes. Depending on the specific channel opened by the neurotransmitter, this binding produces either an excitatory postsynaptic potential or EPSP. This is your gas pedal. It's caused by a net positive cation influx that brings the membrane closer to its firing threshold. On the flip side, we have the inhibitory postsynaptic potential or IPSP, which acts as the brakes. It opens channels for potassium to leave or chloride to enter, hyperpolarizing the membrane and moving it farther away from that firing threshold. Keep in mind, both of these are graded potentials, meaning they vary in size.
Section 3. Calculating the grand potential.
Since individual EPSPs and IPSPs are tiny graded potentials, a single one is almost never enough to trigger an action potential on its own. The receiving neurons' dendrites basically have to act as a computational processor. They take in thousands of competing signals from converging neurons to calculate the grand postsynaptic potential, or GPSP. It's the net balance of all these inputs occurring around the same time at the axon hillock that dictates the outcome. In reality, it can take up to 50 simultaneous EPSPs for the neuron to calculate a net positive and finally fire an action potential.
The cell calculates this grand potential through a process called summation. First, there's temporal summation, which is when a single presynaptic neuron fires in rapid succession, adding EPSPs on top of one another before the first one even has a chance to fade. Then there's spatial summation, where 2 entirely different inputs fire at the exact same time, pooling their excitatory power together. And of course, there's EPSP-IPSP cancellation. If an excitatory and an inhibitory signal arrive simultaneously, they just cancel each other right out.
Section 4. Types of intercellular communication. Up until now, we've focused heavily on fast-acting neurotransmitters, but we really have to distinguish between those classical neurotransmitters and neuromodulators. While neurotransmitters are responsible for lightning-fast signaling to produce those EPSPs and IPSPs, neuromodulators act much more slowly. They are often co-secreted neuropeptides, and they don't cause direct electrical potentials. Instead, they produce long-term modulation of the synapse. They can depress or enhance activity over time, which links them directly to long-lasting events like learning, memory, and motivation. Stepping back, we actually have 6 main modes of communication in the body. You have direct, local communication, like gap junctions that physically link the cytoplasm of 2 cells, or paracrines that are secreted for signaling just in the immediate neighborhood. But if the body needs to send a message across a massive long distance, it turns to hormones and neurohormones. These are secreted directly into the bloodstream by endocrine glands or neurosecretory neurons, traveling far and wide to find target cells that are equipped with the matching receptors.
Section 5. Signal transduction and receptors.
But how does a target cell actually hear the message it receives? This is signal transduction, the intricate process where an extracellular messenger's signal is physically converted into an intracellular response. And it depends entirely on the messenger's chemistry. Lipid-soluble messengers like steroid hormones can just slip right through the cell's lipid membrane like they have a VIP pass, altering gene activity directly. But water-soluble messengers, they are physically locked out. They have to rely on surface receptors, which use either direct chemically gated channels like we saw with EPSPs or indirect G-protein-coupled receptor cascades.
This G-protein cascade is absolutely fascinating. The first messenger binds to the receptor on the outside of the cell, which is coupled to a G-protein snaking through the membrane. Binding activates that G-protein, causing a subunit to shuttle across the membrane and activate an effector protein. This effector then generates an absolute explosion of a second messenger inside the cell, like cyclic AMP. It's such an elegant cascade. One external message triggers a massive internal chemical reaction without the original messenger ever stepping a single foot inside the cell.
And here is exactly why this matters so much for modern medicine. These G-protein-coupled receptor cascades, or GPCRs, provide the therapeutic targets for nearly half of all modern prescription drugs. Because these pathways are incredibly specific, pharmaceutical therapies for everything from asthma and hypertension to pain management are designed specifically to either stimulate or block these exact secondary messenger cascades. Which perfectly leads us into Section 6, Drugs, Diseases, and Synapses.
It's wild to see just how sensitive our synaptic machinery really is. Take SSRIs, for example, which are commonly used to treat depression. They work by blocking the presynaptic reuptake of serotonin, allowing it to stay in the synaptic cleft longer, which boosts mood. Cocaine does something remarkably similar, but with dopamine, blocking its reuptake to create prolonged, intense pleasure that ultimately leads to severe addiction. And on the dangerous side, tetanus toxin works by preventing the release of inhibitory GABA. Without that crucial inhibitory brake signal, the result is unchecked excitatory signals, leading to terrifying, uncontrolled muscle spasms, commonly known as lockjaw.
It truly makes you wonder, what GPCR cascades driving half of modern medicine? What undiscovered therapies lie hidden in our cell's second messenger pathways? Thank you for tuning into this lecture. If you found this breakdown helpful, please make sure to like, subscribe, and drop a comment below with any questions or topics you want covered next. See you in the next explainer.